Yes, indeed.
Yes. Okay, excellent. So if you want to get your slides up on the screen, I'll give you a brief introduction, then we'll switch over to you. So now we have Dag Harmsen, who is an MD specialized in clinical microbiology and infectious diseases epidemiology. He holds a professorship at the medical faculty of the University of Münster in Germany. So Dag has a stellar career in understanding bacteria on a population scale. He published the first worldwide study of whole genome gene by gene analysis, which we now call cgMLST today. He is responsible for a number of microbiology bioinformatics protocols such as SPA typing for MRSA, MIRU for VNTR in tuberculosis. He is also the co-founder, shareholder, and co-managing director of Rydom, which is a company that develops bioinformatics software such as StaffType and SexSphere, which and these tools are used for clinical and public health microbiology sequence analysis. So Dag, very pleased to have you here today. And we're going to hear from you about cgMLST Revisited, Evolvement and Challenges.
Yeah, hello, good evening, everybody from Germany. Pleasure to join you. It's in fact my first online conference until now. I really enjoyed it, and I hope your enjoyment will stay the same during my presentation. Yeah, thanks to kind introduction by Nabil, I don't have to add many things. I'm not a developer, as most of you, therefore my talk will be a little bit different. However, we develop in the academic environment, but also, as mentioned, professional or commercial software tools, and this is my commercial disclosure for today. However, I hope there will be no commercial bias during my presentation. What I'm going to present you is sort of historic personal view, how I believe at least how cgMLST evolved. I have seen several people also contributing to this evolvement heavily. Joao at Taboda, I've seen Nabil, and probably Keith Jolly is also in there. Therefore, feel free to correct me or to comment. So my personal first encounter with next generation sequencing was nearly 15 years ago during an ASM general meeting in Atlanta. And there was a booth by 454 and Roche introducing the 454 sequencing technology. At that time, I didn't understand anything about, didn't understand emulsion PCR, so it was a little bit expensive. So I decided I don't have to care about this, something for basic research. But as always, things developed more quickly. And we had first encounter with next-gen data in 2008. My colleagues in Münster, Alexander Mellmann and Helge Karch, had a collaboration with Bosch U people, Shana Leopold and Phil Tarr, on a PNS study regarding evolution of E. coli 157. And here they use next-gen whole genome sequence data to come up with a core genome SNP sort of shared concatenome SNP analysis. And that was around end of 2008 when Alexander Mellmann and I decided, oh, we were trained up with MLST, we were forced it, we were doing MLST for more than 10 years already. Why not using allele calling genome-wide instead of SNPs? And at the well-known Applied Bioinformatics and Public Health Microbiology conference, I met Keith Jolly, and he showed me for the first time the prototype of what later would become BixDB, and therefore I learned pretty early that others were thinking along the same line. So the next step in cgMLST evolvement was certainly the publication of BIXDB, Martin Maiden and Keith Jolly, University Oxford. That was November 2010. However, they didn't, in this publication, this tool is, they described this, suited for genome-wide gene-by-gene allele calling. However, during this publication, they applied it for from concatenated sequences using SNP calling. Therefore, I believe at least that our STEC O104 paper half a year later, 2011, is in fact the first core genome MLST publication. This was again together with Alexander Mellmann and Shana Leopold from Bosch University, was in the meantime also in our lab working together. We made a quick analysis using BIXDB at that time, and we got a lot of attention due to the fact that this was also the first publication using the OnTorrent desktop PGM machine. However, I personally believe on the long run, the most biggest impact came from the fact that this was sort of core genome MLST allele calling. It wasn't called like this during that time. There was in fact no name. Internally, we called it super MLST or MLST on steroids or something like this. The next step that was important for us was we were lucky enough to get a 6 million euro funding from EC with a project called Pathoenzyme Trace that started beginning of 2012, and due to our scientific advisory board members, we have links to ECDC via Rene Henriksen to DTU, CGE, and also very important, there was already at that time links to what later became a reader via Gary van Domselaar, who visited us 1-2 times during that project duration in Europe. This is the people involved. You see the Oxford people, Münster people, our coordinator, Stefan Niemann, a TB guy. And we had, in addition, associated partner, my colleague in Münster, Alexander Mellmann, and last but not least, certainly Joao Carricho, who contributed mainly with his ontology work to this project. During the course of this project, several public influential papers came out from members of the consortium. This is the ribosomal MLST paper, April 2012, published by Keith Jolly and colleagues. 1-2 years later, they also added R-MLST sequence types. That was not initially, so already a glimpse of a whole genome, gene-by-gene allele calling. Next, at that time, BIXDB was the only available software tool. We came out next with our commercial Zexphere tool, with this Nature Biotech publication where we compared different sequencing machines. At that time, we didn't use the term, still didn't use this term cgMLST. We introduced a new term, MLST plus. And we did this by intention to avoid the term core genome, as core genome genes are here from us, and also you have just heard it from Enterobase, are determined from DNA with rather high similarity values, in contrast to the traditional core genome definition that is done on protein level, bidirectional, and with rather low similarity values. That might, by the way, explain also the differences in the number of core genome MLST depending what approach you take. It took only half a year longer, and Martin Maiden came up with very widely read review. He called this, and that it's the title of my today's talk, MLST Revisited. And here he introduced the terms whole genome MLST that was in fact already used first time April 2012 by Sam Shepard. Sam was at that time member of the Oxford Martin Maiden group, and he introduced for the very first time the term core genome MLST. From my understanding of this paper, cgMLST at that time meant for the authors not a fixed set of loci, but only shared loci of selected isolates on the study. So not yet a fixed scheme as we know it nowadays. This is publication, from my understanding, first original publication using cgMLST, and that was done together with Stefan Niemann half a year later in April 2014 with Mycobacterium tuberculosis. Next, there was a short, Seamin mentioned this, we organized in Oxford sort of cgMLST harmonization workshop, and this was in March 2015. Many, if not all, stakeholders were involved, and there was at least a sort of informal agreement that a cgMLST scheme is a fixed and agreed upon number of genes for each species or group of closely related species that is going to be at least the minimum denominator for analyzing whole genome shotgun sequence data for surveillance purposes. Yes, we tried during this meeting also to harmonize. It was already on the horizon that there would be competing schemes for the same species, but somehow we didn't manage this because there were always between the different pipelines that were popping up slight differences in allele calling procedures that made it also technically challenging to harmonize the whole procedure. And that is where we still are. Next, there was Sister at Tabodar and the Canadian IRIDA colleagues that published in January 2016 Sister, Salmonella and Silico typing, mostly, I believe, used for genomic serotype prediction, but however, they also introduced here, from my understanding, for first time, a core genome MLST approach within IRIDA, and I just learned there's now even another approach in IRIDA implemented. Next, came up the second commercial software tool, Bionumerix 7.5, published first time together with CDC colleagues in March 2016. They were part of the PathoEngine-Trace consortium. Martin and I had a little bit hard time to convince them. Initially, they were a little bit like FDA and NCBI, initially more on the Kamer side, and it took them a little bit longer until they adopted core genome and whole genome MLST. And still they have, even nowadays, they support both approaches. However, they put more emphasis on the whole genome MLST than, from my understanding, than on the cgMLST approach that is, from my understanding, better suited, as pointed out already during the intro-based presentation, as you here always compare same against same. Next, in line in May 2016, CDC made a major publication regarding Listeria outbreak detection. And so that was PulseNet US, PulseNet International followed a year later, Céline Nadon, and we had also in Canada during that time a PulseNet International meeting, endorsed cgMLST, and even NCBI switched during this time together with FDA and the Genome Tracker project from k-MER detection to cgMLST for their pathogen detection pipeline. And next, I'm fully aware that is Enterobase, Nabil, I'm fully aware that you were earlier online, I believe 2016 or 17. I'm counting for all the different services, if available, only the first publication that became available. And Enterobase, it's, as it was later developed, is a very sophisticated system. In general, there's a tendency that the later systems, not so surprising, are somewhat at least more sophisticated than the earlier ones. And finally, there's two new additions. One was already mentioned. This is Innuendo, done by Mirko Rossi, at that time still in Finland, I believe in the Veterinary Institute, and João Carisho and his mostly Portuguese colleagues. And more recently, the Pathogen Watch system. I believe there's still not yet a publication done by David Arnson and his team members. That is, I believe, all systems that at the moment do cgMLST. I hope I haven't missed anything. And for the remaining, I skip this. For the remaining minutes, I will quickly allude a little bit regarding the cgMLST challenges. A major problem, of course, is with the competing schemes. However, this is, as so frequently, not so much a technical challenge, it's more a social challenge. Due to the different pipelines, the various players have their own ambitions, and it's very, very complicated, let's say, to harmonize this. I would focus on two other topics. So you have finally here a long list of numbers, and these numbers are extremely good for computers. That is a computer readable nomenclature. However, an additional identifier of profiles for human communication purposes would be desirable, at least. The challenge, though, is that we are dealing with an expanding, so not a closed nomenclature, and to slightly less extent, we also have to deal with missing data. There was in 2014, 2015, the idea to have a taxonomically, or I would call it phylogenetic nomenclature. This figure is taken from an ECDC expert opinion, but I know also CDC, the PulseNet team had during that time same sorts. So in an ideal case, every isolate sample would have an identifier like this 111, where you immediately see from the name where it is placed in the tree. From my understanding, of course, something like this would be desirable. However, it's impossible for an additive expandable nomenclature system, as there will be always changes in tree. Therefore, I believe phylogenetic nomenclature was in the past impossible, even with canonical SNP for monomorphic organisms, and will be also in the future out of reach. What is possible and we have heard already a little bit about this, is similarity-based nomenclature. With our release of SECsphere, we had from the beginning 2013 what we called complex type CT, not hierarchical nomenclature. This server was only made public, but was always there in 2016. Now that is a little bit busy slide. What we introduced was what I would call informal static founder concept. So if new systems are submitted to the cgMLST system, and the allele distance is less or equal than the CT founder, it's lumped together with this founder. This prevents tailing of complexes. However, the problem, and that is shown here below, is Joao called this is sort of fragmentation. Like MLST, the actual numeric CT value does not express relationship. In contrast to MLST clonal complex that uses a dynamic founder concept or public health SNP addresses, we used right from the beginning CT thresholds that are species specific. The problem you see here, depending, that's an example of the entry order, you get samples lumped together in different complexes. So we have a stable nomenclature, however, we have fragmentation. There's another system, and this slide is courtesy to Tim Dahlman from Public Health. That was very influential for my research, first published in 2016. It's the SNP address. You all probably have heard. They have 7-digit numbers separated by full stops with different levels. So 0 SNPs different, 5, and so forth. And this gives an address, and they use similar like Enterobase, a hierarchical single linkage clustering. And just to give you an example, let's assume the SNP level is 5 thresholds. You have a group, all you don't have a founder here. All isolates that are less equal or less than 5 distant are brought together, and therefore elements at the opposite ends of a cluster may be much farther away from each other than this threshold. This phenomenon is well known as tailing. The other problem is if you have a merging of complex, you must have a tie-breaking rule, sort of, either, and thereby the nomenclature is not stable. Tie-breaking could be similar like Enterobase, that you use the lowest number of the SNP level or the larger one. Anyway, somehow it looks like that this is the winning system, because BixDB and Enterobase both with always same thresholds, and Innuendo with species-specific thresholds implemented a similar hierarchical single linkage clustering allele nomenclature approach, and this is very popular services. Both approaches are not fully satisfying, but at least I myself cannot imagine of a better approach, and therefore we probably have to live with this limitation. I would like to end citing Dag Harmsen. You know, Trump is not happy with WHO, and Dag Harmsen was the leader of the United States. Nations There's a famous quote: "It has been said that the United Nations was not created in order to bring us to heaven, but in order to save us from hell." The same could be said to WHO, by the way. And I'm saying it has been said that cgMLST was not created in order to bring us to heaven. It's by far not perfect, especially this human nomenclature thing, but it's better, but in order to save us from hell. And by this, I'm happy to take questions. Thanks indeed.
All right, thank you, Dag, very much. That was excellent. Joao has said in the comments, thanks for this perspective. Now I feel kind of old.
Yeah. You have seen me. I'm the old white man.
But yeah, so I have a few questions that people have put in the chat, and I'll put them onto you. One of the questions is, how is missing data defined in SecSphere? And how is it handled, for example, when a minimum spanning tree is calculated?
I know that you have a special system in Enterobase.
I've not asked someone else.
Yeah, Manu was involved with this. I believe we have a simpler approach. The default setting in SecSphere, at least, is that we don't use missing data in the pairwise comparison if it is missing in one of the two strains. This is not perfect, as you know. There's whole books and statistics dealing with missing data, and there's tricks and ways to handle them. You cannot solve it fully, but in our hands, it usually works pretty nice, because if you have a denominator, let's say with Salmonella or E. coli of 2,500 or 3,000 alleles or genes, and a fewer ones are missing, it's usually robust enough to compensate, in our hands at least, for missing data.
Okay. So I had one question, which is about cgMLST. I was wondering, so there's been a lot of work on cgMLST, obviously, but is this going to be accepted as a standard approach in public health? And what does the ECDC think of it?
Difficult. From my feeling, I believe it's accepted in the meantime, not from the beginning, but around 2014, 15, for clinical and public health microbiology. And the reason is that CDC took over this concept with PulseNet and PulseNet International, and by their sheer definition power, nearly all labs and countries had to follow. The food people are still, from my feeling, a little bit reluctant. FDA is, although NCBI is applying core genome, MLST is internally with their pipeline still using SNP calling. As we have heard, there will be a time when they have a problem with scaling and sheer compute power. However, they use more and more often also core genome MLST, I know, and there's also a number of publications. The other influential food agency is DTU in Denmark. They were always strong KMER believers, I would call them. They have more recently, I believe, even implemented core genome MLST. I'm not sure, but from my feeling, they are still a little bit reluctant. So food is probably not yet decided. And regarding ECDC and EFSA, we have Mirko Rossi from EFSA now online. He can comment probably better. from my understanding, at least ECDC is already accepting, I believe, for Listeria, core genome MLST data, and they want to extend this soon, or already doing this for the other zoonotic organisms. And as food arena is a little bit more regulated, I believe EFSA will join them later, but I'm not absolutely sure. It's a little bit political.
All right, so I think we'll close with one more question. What is the future of cgMLST then, and what is the most important element of it that we should be focusing on?
Yeah, I believe, although we have, I stressed this, and it was mentioned before, we have this competing schemes, and that is a little bit ugly. Martin Maiden is always saying, you have the same, you know this, with MLST and E. coli. I'm aware at least about 3 schemes. There will be, or is already, a sort of, Martin calls this a Darwinian selection of schemes. So finally, the winner will take it all, like the Achtmann E. coli scheme, because he had simply most submissions. Not necessarily the best scheme, you can argue, but was most widely used. And I believe we will see similar with this competing schemes for core genome. And obviously, there's a limitation in discrimination power, and there's nothing against putting on top SNP calling or whole genome. However, it's the benefit, it's so obvious, you can, between laboratories, pretty easy exchange results and exchange data with your colleagues, low volume data. So that's the benefit, I believe.
All right, and I think that's a good place to close. I'll post a link to the Discord in the chat. If you haven't, please join us. We will be around in the virtual pub with, there's already a bunch of people at the drinking table. Please do join us. We'll continue the discussion. There's a hearty discussion in the Discord right now about Dag's talk. A lot of positive feedback for you, Dag, and a lot of positive feedback for all of our speakers. I hope all of our speakers today appreciate that there's a lot of people watching out there. I think we had maybe 114 at the peak, all listening in, and so far a lot of positive feedback. So thank you very much to all of our speakers. And that is the end of the presentations for today and the end of the Zoom session. Please switch over now to the Discord, where we can continue the discussion, socialize a bit, and get to talk about some of the different elements we've raised today. All right, thank you very much. From me, I will see you this time next week for the next session. So thank you very much, everybody. How do I turn it off?
Yes.